Hello again, and welcome to part 3 of this 3-part lecture series on viruses, viroids, and prions. Okay, so we just finished up talking about the lytic cycle, which causes the bursting, the lysis of the host cell. Host cell dies. We're going to look at the lysogenic cycle, and I have this as the prophage DNA, which is inserted phage DNA. Prophage means pro means pre, so this is before they're actually becoming a phage. That gets incorporated into the host DNA. So all the instructions to make the phages are incorporated into the host DNA, but it doesn't go through the biosynthesis, the maturation, and the release part in the lysogenic cycle. It just gets replicated over and over and over again. The host cell does not die, and every time the host divides, so does the prophage DNA. So the DNA becomes actually inserted into the host cell DNA, and now all the instructions that are necessary to make the phage are embedded into the host cell DNA, and every time it replicates, the host cell that is, the prophage DNA replicates along with it. And so what happens here, and you look at this and be like, what the heck is going on? So on the left, there's the lytic cycle. We just talked about that. And then on the right is the lysogenic cycle. So start up at the top left there where it says the red number 1. Well, the number 1 is white, I guess, and the red circle around it. The phage attaches to the host cell, injects its DNA, and then you have the bacterial chromosome, that's that blue oval. And then you can see in the middle they're side by side. Well, if you continue that circle down to if you keep going down to the left, we enter the lytic cycle, where you get biosynthesis, maturation, and then release. But follow it like a figure 8. Go to the middle picture and then go down to the lower right. The prophage DNA is now integrated with the bacterial chromosome in that DNA. Then every time the bacterial cell reproduces, the prophage DNA reproduces right along with it. And that says many cell divisions, so again and again and again. Occasionally the prophage, the DNA, may excise or remove itself from the bacterial chromosome by another recombination event, initiating a lytic cycle. So then it could go down into the middle and then go over to the lytic cycle. And if you have a whole bunch of bacterial cells that have the prophage DNA with them, replicating, and dividing, and then they enter the lytic cycle, that is disaster for the bacterial cells. And this is how prophages do their damage. That's why they're considered, I guess you could say, the deadliest thing on Earth, because they kill bacteria by the bajillions every single day. So some of them are going through directly into the lytic, and some of them are going into the lysogenic, and then billions of bacteria have the prophage DNA, and then they exit the lysogenic, go into the lytic, and boom, out come more of the bacteriophages. And again, remember this, and I come back to phage therapy. Sorry, I'm a little, what do I say, a little biased towards phage therapy. Did you notice that? This is what happens. It's a lytic cycle. So they inject their DNA, they get inside the bacterial cell goes boom, and then new phages go out and infect other bacterial cells that are infecting you. And then eventually that infection clears up rather quickly, much faster than with antibiotics. Typically, not always, of course everybody's unique, but that's typically what is found, that the infection clears up way faster than with antibiotics. So anyway, sorry. Off on the phage therapy rabbit trail again. So what about animal viruses? I said in part 2 that we talk about that. Well, hey, it looks pretty similar, doesn't it? We have attachment, penetration, we have an additional one called uncoating, then biosynthesis, maturation, and release. So again, it attaches. Virus attaches to cell membrane, and some individuals it can't, and that makes you immune to it. So how are you immune to it? You do not have the viral attachment sites. So some people have immunity to what's known as a parvovirus, for example, which causes illness and disease within people. Well, they don't have the attachment sites for those viruses, so those viruses never gain access into their cells. If there isn't an attachment site, the virus cannot attach itself to the cell and enter it and then cause some kind of an infection. And so you have innate immunity, meaning you've been born with that immunity because you don't have that cell membrane attachment site. Why don't you? It's genetic. Thank your parents for that. It was very nice of them to give you that gift. Now, that doesn't mean that they have immunity to it as well. This could be a recessive gene. that got passed on to you. Or it could have been a sporadic or a spontaneous mutation in your cells and you didn't produce that attachment site, and so you have developed immunity against it. Will you pass it on to your children? Maybe you will, probably not, but you might. So this is all a genetic factor. Penetration, meaning getting into the cell. Now we're talking about eukaryotic cells. So bacteria do not carry out pinocytosis or membrane fusion, so they end up getting into the cell that way by fusing with the membrane. Then there's uncoating. The nucleic acids become separated from the capsid. Remember, the capsid is the protein coat that surrounds the nucleic acids. That capsid gets separated. The nucleic acids are released inside the cytoplasm, and then it heads down into the nucleus of the cell. Gets the nucleic acids from the host cell, then uses those nucleic acids to make more of themselves, gaining access to the host cell nucleic acids and the host cell enzymes. Then they can undergo biosynthesis, making more and more of them, maturation. The nucleic acid and the capsid proteins, they assemble themselves together. The capsid envelops itself around the nucleic acid, and then there's release by budding, in which enveloped viruses, that's how they leave the cell, because they have a lipid envelope around them. Now imagine you're looking inside the cell in the cytoplasm. That lipid envelope merges with the cell membrane on the inside of the animal cell, of our cell, say for example, and then it buds out of it. Usually that doesn't kill the host cell. Why is that of advantage to the virus? Well, you don't kill the host cell, it can be reinfected again. Or rupture, these would be non-enveloped viruses, this kills the host cell. So they don't have a lipid envelope that can merge with the phospholipid bilayer of the cell membrane, so they have to get out another way. How do they do it? They release enzymes, burst the cell open, and that kills the host cell. And there's an interesting virus video my college students could click on. It's very rudimentary, very simple, but it's a decent video, and it has some good animations of how the viruses are able to do this. You can see how there's a picture of budding on the left, and then you can see the buds are all those little bumps that are on a lymphocyte, HIV-1 buds on a lymphocyte, and then the bottom picture you can see they have arrows. In case you missed those little bumps there they are. So they are budding out through the cell membrane, not necessarily killing the cell, usually not killing the cell. The cell remains intact. What about viruses and cancer? Well, that's a good question. Anything that can alter cellular DNA, oncogenes, has the potential to make normal cells cancerous. Do viruses alter cellular DNA? Of course they do. Several types of cancer are known to be caused by viruses. At least, as far as we know, 10% of all cancers are caused by oncoviruses. The relationship between viruses and cancer, first demonstrated in 1908. Chicken leukemia. So they were doing some research with chickens and leukemia, and they sent this cellular fluid from tumor masses from chickens and cancerous cells. They had this kind of cellular soup. They ran it through a filter, thinking they were going to filter out what was causing the leukemia in this filter. Then they took it, and I assume injected it into healthy chickens. Well, they developed leukemia as well. Now, of course, we didn't know they were viruses particularly at that time, but we knew this was some kind of, remember, the word virus comes from Latin for poison or toxin, and so they thought it was some kind of poison that was in these chickens that were dying from leukemia, and it was being passed on to others where they thought they might be able to filter it out. Nope, they weren't able to, and that was a testament to the incredibly small size of viruses. So scientists started to realize they were dealing with something that was incredibly small. Oncoviruses are viruses that transform normal cells into cancerous ones, and so we call these cancerous cells transformed cells. They have increased growth. They have loss of contact inhibition, meaning normally when cells contact each other, they stop dividing because they send cell signals to each other that says stop the cell cycle, and so they stop dividing. But transformed cells say, I don't have any personal space. You can divide and multiply as much as you like. Social distancing? No way. So they're going to pile up and form these tumor-like masses. And viral specific T antigens. What are they? Antigens that disrupt the normal cell cycle. So they put the pedal to the metal and mitosis just proceeds uncontrollably. The genetic material of oncogenic viruses becomes integrated into the host cell's DNA. It's similar to lysogeny. Like, remember the lysogenic cycle, where that DNA becomes integrated into the host cell DNA? That's what they're doing. And so as a result of that, it's transforming those cells, and it's causing them to multiply and divide rapidly rapidly and recklessly, with no contact inhibition, increased growth, high-speed division. And then, as a result of that, also what comes on later is angiogenesis, where it starts directing blood supply to this tumor so that it can feed the tumor, this incredibly metabolically active mass of growing cells. And so they start leaching blood from whatever organ or tissue they're growing in. The organ or tissue starts to lose nutrients, and those cells begin to die, and the cancerous tumor cells, the transformed cells, they thrive as a result of that. So they become basically a leech, taking the nutrients from the normal tissue, from the normal cells, and hogging it all to themselves. This is just oncogenic DNA viruses, ones that we know cause cancer. These are DNA viruses, so you can take a look at those. It's just a few here that I listed and I got from that website there. And some of the cancer, so the middle one is the disease. What disease does it cause? And then if they do cause cancer, if they're oncoviruses, what cancer do they cause? Here are a few RNA viruses that we know are oncogenic, retroviruses that are oncogenic viruses. Okay, what's the difference between latent and persistent viral infections? Well, a latent viral infection, virus remains an asymptomatic host cell for long periods of time. So your host cell has the viral DNA in it, but the host cells are asymptomatic. They're not showing any symptoms of it. There's no cytopathic effects, but they are there. The viruses are there, and they stay with you. So, for example, cold sores, if you get HV1, human herpes virus 1, develop cold sores, these guys stay latent in what are called the trigeminal nerve ganglia. And if you know your anatomy, ganglia are just masses of neurons, and cold sores will remain in those ganglia. Shingles, that's from the chickenpox virus, which remains latent in dorsal root ganglia of the spinal cord. So on the backside, the dorsal root, and where the masses of neurons and bundles of neurons are, that's where those viruses, which chickenpox is a herpes virus as well, that's where it remains dormant. And oncogenic viruses will do the same thing. Asymptomatic, and then they begin causing transformed cells in cancer. So you may have had these viruses for decades, and then they reappear later, can be reactivated by immunosuppression or some other factors that we just aren't sure of, especially with oncogenic viruses that are latent ones that are causing cancer later. But immunosuppression seems to be the thing that is the trigger. So I had chickenpox when I was a kid. I was young and recovered from that, obviously. And then decades later, how many decades? Oh my, about 46 years later, I developed shingles. I just had it this past spring, and it's painful. So those chickenpox viruses, those herpes viruses, were remaining latent in my dorsal root ganglia, and then because of, I don't know if I'm immunosuppressed or not, or if it was just stress, or I don't know what caused it, but the shingles reappeared. And instead of being kind of appearing generally all over the whole body as these the little vesicles that chickenpox produces, the shingles is in a very limited area because they travel down one dermatome, which is a dermatome is a region of skin that's served by a particular nerve that's coming out of the spinal cord. So shingles is much more localized than chickenpox is. Because it's staying latent in a very specific spot on the spinal cord. Mine appeared right around my chest and my back, my upper back and my chest. and it's painful. And typically only presents on one side because it usually stays on one side of the spinal cord, but sometimes they're latent on both sides, and you get shingles, vesicles on both sides of the body, which I would hate. So I feel badly for people who get it on both sides. You get double the pain, double the irritation and sensitivity from it. So they're latent, they're hiding, and then they will reappear later. Persistent, or what we would call chronic viral infections. So disease progresses over a long period, generally fatal, and the disease presents typically years after the initial viral infection, often caused by another virus that will lead to it. So, for example, measles and fatal encephalitis. About 1% of all measles cases develop fatal encephalitis decades later in life, where the brain starts to swell uncontrollably and it's fatal. HBV and liver cancer, hepatitis B virus and liver cancer years later, human papillomavirus and cervical cancer. So it's a chronic, persistent, doesn't go away viral infection. It's not asymptomatic like latent is. It progresses and there are symptoms, but it does usually present years later. Prions. Prions are a mystery. Prions are proteins. They have no nucleic acids in them at all, but they're able to replicate. And much of what we know about prions, if you watch any video about prions or read an article, a good article, scientific one about prions, not some other junk that's on the internet, but one from a good reputable source, the terms that are used is it is theorized, it is hypothesized, it is speculated. Well, they typically don't use the word speculated, but hey, it means all the same thing. In other words, what they're saying is, we're not sure what's going on here. We think this is it, but we really don't have any idea. They were first explained in 1982. Now, prion diseases have been around obviously much longer than that. But a scientist named Stanley Prusiner, he first explained them in 1982, won the Nobel Prize for explaining them as a result of his investigation and explanation. But even since then, we're not sure what's going on with these prion diseases. They're inherited and transmissible by ingestion, transplant, and surgical instruments. So you say, well, surgical instruments, they become autoclaved. Well, they survive autoclaving. Prions are, once they become an infectious prion protein, they are incredibly stable and resistant to denaturation. Remember, proteins can become denatured, or their shape can change. And when the shape changes, or when the shape shifts, the protein is now no longer functional. Well, prions are so incredibly stable for reasons scientists have no idea why, they can survive an autoclave. Typically, in order to get rid of them, we have to use enzymes, concentrated sodium hydroxide, and autoclaving, putting them all together to try to destroy these things. They are all fatal. There are 5 prion diseases currently that we know about. All 5 of them are 100% fatal. There's no treatment for them, and there's no cure for them. If you are diagnosed with a prion disease, you are going to die. Nobody has survived one, and we don't know what to do with them in order to treat patients. So all that's really given is palliative care, just make them comfortable, and so on. There was one instance of an Italian guy, can't remember his name, but he had a prion disease called fatal familial insomnia. Look it up. It's crazy. Very, very, very few people have it, but he did. And he knew that he got it. He was diagnosed with it. And typically when you're diagnosed with a prion disease, you're dead within about 9 months, sometimes 6 months. It's a rapidly progressing disease. So he was diagnosed with it, and he said, okay, I know I'm going to die, but what I want you to do, and this was over in Italy, the University of Bologna? Sorry, not Italian. But anyway, he said, look, I'm going to die. I want you to, keep me here. And as I start to slowly degrade and die and lose my mind, I want you to research on me. And then when I'm dead, take my brain, autopsy it, cut it up into pieces, do whatever you need to do to understand this disease. So he donated himself to research. He knew he was going to die. There was no hope for him. And so I think it was a very noble thing that he did to allow him to be researched, and you can watch a video on it. He was completely incoherent. He had no idea what he was doing during the last weeks of his disease before his death. And then they took his brain, and they learned a lot from it. So you can check that out, fatal familial insomnia. They're resistant to just about everything. Many are believed to be inherited as well. Creutzfeldt-Jakob disease is probably the most known. There's a classic Creutzfeldt-Jakob, meaning like this is the one we know and this is the way it's usually transmitted. But then they discovered there's a variant of it. So it's a CJD with a V in front of it, variant Creutzfeldt-Jakob disease. There's one caused by cannibalism. So there was a tribe in Papua New Guinea, a few tribes that practiced ritualistic cannibalism, and they would consume the brains of their deceased relatives. Um, their deceased relative died because of a prion disease called Kuru. They ingested their brain tissue that had the prions in it, and then they developed prion as a result of that. In Pennsylvania, we have chronic wasting disease among the deer population. Um, they say it can't be transmitted to humans. Apparently it's not, but it raises some concern. They said mad cow disease could not be transmitted to humans, but it appears that there were some cases in which humans did ingest beef with that came from cows that had the prion disease, mad cow disease. They consumed that beef and then they developed a prion disease as a result of it. There's a lot of speculation on that, a lot of disagreement, but it kind of looks like that was the case. So how are they designated? PRPC is the normal cellular prion protein. We have them on our cells. Our cells have these proteins that are there and they're carrying out functions. And then what happens, for reasons we don't know, it develops into what's called a PRPSC, a scrapie protein. They call it scrapie because that was the disease that was being studied by Stanley Prusiner, the guy who discovered it, or not discovered it, but it described and explained it in 1982. He was researching scrapie disease in sheep. And so that's why it's designated the PRPSC for the scrapie protein. It accumulates in brain cells, forms these plaques, similar to like plaques and tangles that you see in brains of people who have died from Alzheimer's disease. And so there may be a relationship between prions and Alzheimer's and even Parkinson's. We're not sure. So they're doing a lot of research with those diseases and seeing if there's some kind of association between them. The plaques are used for post-mortem. They don't appear to be the cause of cell death. And so whenever they do the brain autopsy or the brain dissection, they see these plaques. And then the cause of death then is known, oh, it was a prion disease. For somebody who died for unexplained circumstances or unexplained reasons, when an autopsy is done, they look at the brain tissue and they see these plaques there, and then they can tell it was a prion disease. There is still much to be learned about them, and that's an understatement. Fortunately, there are only 5 of these diseases in a very small portion of the population, of the human population globally, has had prion diseases. But they are they spread within the individual. So what's going on with them? Could they spread to other people? That's what we have to be very careful about. They're infectious proteins. They're infecting other ones, and they're causing these prion diseases to rapidly and uncontrollably spread within a victim. Can they be spread to other humans as of now? We say no, but we need to be careful, and so this is being researched. The microscopic holes are characteristic. This is a brain tissue under the microscope. Those holes should not be there, and so we call them spongiforms. So, because it looks like a sponge, it's got holes all through it like a sponge. It gives the tissue a spongy appearance. This is just a quick graphic here on how they spread. So the green boxes, those are supposed to be the normal cellular proteins, and then in step number 2, we have the scrapie protein. You can hear the dark side music, bum, bum, ba, bum, bum, ba, bum, bum, ba, bum. And then what the scrapie protein prion does is it converts the normal cellular prion protein and converts it into a scrapie prion protein, takes it to the dark side, and then they will all go and infect other ones. The cell tries to ingest these infectious proteins, the infectious prions, but it does nothing because the enzymes in the cell are not capable of doing anything to these prions. So they continue to, quote, survive. But remember, they're not living things at all. They're not even close to it. I mean, viruses are, okay, maybe they are. Prions, no way, because they're proteins. And if you are a deer hunter, check out Chronic Wasting Disease, a very good website. The best one that I found is cwd-info.org. It gives all kinds of info on chronic wasting disease, and if you are a deer hunter, you should inform yourself on this disease, and that's a great website to go to. Plant viruses and viroids, hey, there's not too much to say about that because plants are generally protected by their cell wall. The cell wall is a very stiff, rigid, sturdy, thick structure that protects plants very well. It's very effective. So plant viruses enter through wounds or via insects that suck the plant's sap. So the insect is infected with the virus. It goes to take the sap from the plant, drills its way into the xylem, the vascular tissue of the plant, and whenever it does that, it's releasing saliva, and in the saliva are the viruses, and then that gets into the xylem and the phloem of the plant tissue, and then can circulate through the plant, and then that can kill it. Viruses can multiply inside insect cells, and so because of that, then they can spread it from one plant to another. It's kind of like, how malaria is spread by the mosquito. The mosquito secretes saliva when it drills its little needle-like proboscis into our skin, and when it releases the saliva, it also releases the protozoa. Well, insects are doing the same thing to plants. Viroids are infectious pieces of RNA. It ends in oid, so it means it's like the prefix. So these things are like viruses, but they're not. Probably the most infamous one is the one that causes potato spindle tuber disease. It's responsible for millions of dollars in crop damage. So these are just little infectious pieces of RNA. They're usually, we think they're introns. They seem to be, and those are the interrupting or the non-coding segments of RNA that do not code for proteins. These viroids are very similar to those. So they're just infectious pieces of RNA that can be spread throughout a plant and from plant to plant. So it basically does that mottled appearance to the leaves. The leaves can't photosynthesize, and then the plant dies as a result of this infection. Okay, that's it for this chapter on viruses, viroids, and prions. So hopefully you enjoyed this, that it was of benefit to you. I sure enjoyed lecturing about it. This is good stuff to talk about, especially phage therapy and oncolytic virotherapy. But I said I wouldn't talk about that anymore. So thank you for watching, and I'm done.